With great joy and immense exultation, I feel privileged to extend my warm welcome to all presented here for the inauguration. Every morning was a cheerful invitation to make my life of equal simplicity, and I may say innocence with nature herself. Prayer is not a discuss, it is a form of life. The life with God, that is why it is not confined to the moment of verbal statement. Now start prayer song.
God will make a way where there seems to be no way. He works in ways we cannot see. He will make a way for me. He will be my guide. Hold me closely to his side. With love and strength for each new day, he will make a way. He will make a way. Oh, God will make a way where there seems to be no way. He works in ways we cannot see. He will make a way for me. He will be my guide. Hold me closely to his side. With love and strength for each new day, he will make a way. He will make a way.
Thank you, friends. Surely the immutable law of the universe can teach more impressive and exalted lessons than the holy books of all the religions on earth. Now start holy books reading.
Holy Bible. The Gospel reading is taken from St. Luke chapter 11 verses 21 to 27. At that time, Jesus said, I thank you, Father, Lord of heaven and earth, because you have hidden these things from the wise and the intelligent, and have revealed them to infants. As Father, for such your gracious will, all things have been handed over to me by my Father, and no one knows the Son except the Father, and no one knows the Father except the Son, and anyone to whom the Son chooses to reveal him. The word of God. Praise to you, Lord Jesus Christ.
The words from the book of Bhagavad Gita. You came here empty handed and you leave empty handed. What's yours today belongs to someone else yesterday and will belong to someone else tomorrow. So don't think much. Whatever you do, make it an offering to me. The food you eat, the sacrifice you make, the help you give, even your sacrificing. Whatever happened, happened for the good. Whatever is happening for the good. Whatever will happen, will also happen for the good. A famous quote by Lord Krishna in Bhagavad Gita, if you don't fight for what you want, don't cry for what you lost. Nothing depends on lucky, everything depends on work, because even lucky has to work. This is the word of the God, thanks to be the God.
Reading from the Holy Quran. أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم اللهم إني على ذكرك وشكرك وحسن عبادتك. Oh Allah, help me remember you to be grateful to you and to worship you in an excellent manner. These are words of Lord, thanks to be God.
Thank you, friends. It's a glad welcome we bring to you this morning, filled with desires, hopes, and dreams we all share. I'm very happy to invite Ms. S. Sulochana, Assistant Professor, Department of Biochemistry, to the deliver welcome address.
Good morning, one and all present here. I extremely honored to have the chance to address you in this prestigious moment. Beginning a day with a smile makes us cheerful throughout the day. Let us begin with smile and make this day cheerful one. I welcome you all this morning with a cheerful smile. The one act as a pillar of support and strength with a clear vision and passion, entwined with novelty in all the activities of college in our respected Secretary, Reverend Mother C. Noel Rani, FSAG. Welcome you, Mother, to this auspicious occasion. Is the best supporter of all the challenging ventures taken by our principal, Reverend Dr. E. Glita Sumangali, FSAG. You are leader, you show motherly love, give fatherly confidence, your endless care, guidance and support prove that you are great leader. Welcome you, sister. It's my pleasure to bid a genial welcome to our Vice Principal, Reverend Dr. A. John Diana, FSAG, who has been a strong pillar of support to our college. Welcome you, sister. To be inspired is great, but to an inspiration is an honor. Well, it's suitable for our resource person with his true knowledge, Dr. Santosh Shigamani, Team Lead, Product and Process Control at Varsha Bioscience, Private Limited, Hyderabad, Telangana. A very warm welcome to you, sir. Coming together is a beginning, keeping together is a process, working together is a success. That is a team building. Yes, I am very happy to welcome our convener, Ms. J. Sumati, Head Department of Biochemistry. Welcome you, ma'am. I extend my warm welcome to the coordinators, members, the organizing committee, and my dear students who have put more efforts and hard work, which play a big role on this day to be successful one. Welcome to my dear colleagues and dear students. I extend heartily welcome to various college HODs, assistant professors, research scholars, and the students who have united with us to the light of knowledge. Your presence here is highly valuable. Welcome you all. Once again, it's my glad to welcome each and every one of us. Thank you.
Thank you, ma'am. Leadership is the ability to guide others without force into a direction or decision that leaves them still feeling empowered and accomplished. I'm very glad to invite the respected Reverend Sister Dr. E. Glita Sumangali, FSAG Principal, for the deliver the presidential address.
Thank you, Ms. Lokshana, the Assistant Professor of the Department of Biochemistry, for your words of welcome. Respected Reverend Mother C. Noel Rani, FSAG Secretary, Reverend Dr. A. John Diana, Vice Principal, Ms. J. Sumate, Head and Convener of this webinar, Dr. Santosh Sigamani, Team Lead, Product and Process Control at Varsha Biosciences Private Limited, Hyderabad, Telangana, assistant professors, and organizing team members of this webinar, students and participants from various institutions, warm greetings and welcome to all of you. It is my distinct pleasure to congratulate the convener, organizing members for taking tremendous effort to conduct national level webinar on exploring current challenges in genetic engineering and advanced techniques in medical science. The title for deliberation is quite novel and has great relevance. Genetic engineering holds a great promise to the new generation. A growing number of scientists believe that we will soon be able to engineer and change our gene the way that help us to live longer and healthier. Nothing about our life is natural except the oxygen that we breathe is natural. Everything is an unnatural, man made. We change our environment. Tackling disease and tackling aging is not natural, but that is what we do. We do accept misery and frailty as natural way of life. Many a times we do not accept misery and frailty as natural ways of life. 2000 years ago, we have been shaped by our inventions, agriculturalistic, industrialist, and eventually modern days now digital technologist. All these advances have profound effect on our species, but we haven't stopped there. Biotechnologists bringing about another age of humanity. As generic engineering technology approaches an unprecedented era of human gene editing, the ethical implications are beginning into rise how to balance our desire to advancement of technology and our fears to misuse of this technology. What is the moreover possibility for future generation? Is this an opportunity for human progress or illuminating disaster in the making? We are beginning to reach the point where we can no longer ignore these questions with technological advances and molecular biology like CRISPR that allow for specific gene editing approaches. Many scientists started reviewing that there is strong potential risk to human genetic engineering. I am certain that there would be wide gap between genetically enhanced one and normal ones. Gene modification is a double edged sword. So I'm curious about what today's presenter is going to speak on this deliberating and debating title. With these few anxious thoughts, I wholeheartedly congratulate the resource person for accepting our invitation and to come and deliver his knowledge with us and to inspire our students an insightful explanation to further take up their research on genetic engineering. I also wish to congratulate the participants to have productive deliberations. Once again, congratulations to the convener and the staff and students of Biochemistry Department for bringing the resourceful person for discussion on a very novel title. God bless your effort. God be with you all.
Thank you, sister. The key to growth is the introduction of higher dimension of consciousness into our awareness. Now I invite Ms. T. Valli, Assistant Professor, Department of Biochemistry, to deliver about resource person with pride and joy.
Happy morning and a very warm welcome for one and all gathered here. I feel very proud to introduce one of the resource person of this webinar, Dr. Santosh Sikhamani, MSc PhD. He is presently working as a team lead product and process control, Varsha Bioscience Private Limited, Jiklapalli, Hyderabad. He is of specialization in microbiology, biotechnology, bioactive metabolics, nanomedicine, and biological applications. He has published journals, 5 papers in Springer, 6 in Elsewhere, 4 Tyler and Francis, 1 Wiley Publishers, 1 IJIR 1 PTBR Heralds, 2 Medno, 1 Innover, 1 Having Medical University, 1 PSB Journal, 1 Pelagia Research Library, 1 Scholar Research Library, and 2 American Scientific Publishers. Also, he experts in biological methods, STS page, RNA, DNA, isolation from algae, bacteria, fungi, plants, polymerase chain reaction, thin layer column chromatography, antimicrobial sensitivity test, and antimicrobial assays, antioxidant assays, DPPH, ABS, FAE, hydroxyl sodium. Dear participants, I request you to make use of presence to the and help me in welcoming you. Sir. Now start a session.
Uh, is my voice audible to everyone? Hello.
Yes, sir.
Uh, I thank the principal for the presidential address and for the Valli for a brief introduction. So now we'll start with the presentation. Technical team, can you enable the video? I'm not able to enable the video. I cannot switch on my camera.
No, sir.
அதுதான் மேம் ஆன் ஆக மாட்டேங்குது. இல்லை நான் மறுபடியும் லிங்க்குள்ளே வரட்டுங்களா? ஓகே. I think my presentation is visible to everyone.
Yes, sir.
There is some issue with the video. Can you hear me? Can you hear me? Hello. Can you hear me now? So I'll start with the presentation. Hello. Hello. Can you hear me?
Good morning. Is there any technical issue?
Yes, ma'am. I am not able to connect from laptop.
But you are
So I'll try.
audible. Your screen was visible before, just before.
Yeah, ma'am. Ma'am, I tried connecting it with my phone, and through that only you would have seen my face. So I'll try connecting it with my phone only, ma'am. Just give me 2-3 minutes. I'll connect it on phone.
Yes, sir.
Yeah. sorry, there was a technical issue with the connection. So I'll be doing the presentation through audio mode. I hope the presentation is visible to everyone. Hello. Can you hear me? Hello. Is my screen visible to everyone?
Yeah, visible, sir.
Okay, ma'am. So I'll start with the presentation now. So coming to today's topic, it is exploring the current challenges in genetic engineering and advanced techniques in medical sciences.
Excuse me, sir. Can you maximize your screen,
Yeah,
sir?
ma'am. Yeah, ma'am. It is in full screen only, ma'am.
Okay.
Is it visible?
Visible only, sir, but little bit minimize, sir. I mean, screen 75% only visible, sir.
Actually, I'm sharing through. Hello. Is it visible now? Hello.
Yes, sir.
Okay, ma. I'll carry on. So I
Okay,
hope
sir.
my
Thank you, sir.
voice is audible to everyone. So genetic engineering. Can I start now?
Yes. Yes, sir.
Ah, yeah. So genetic engineering is the manipulation, modification, and recombination of a DNA or other nucleic acid molecules in order to modify an organism or population of organism. So the term genetic engineering is used to refer the methods of rDNA technology that emerged from basic research in microbial genetics. So genetic engineering has led to the production of very important products such as human insulin, human growth hormone, and hepatitis B vaccine, as well as to the development of the genetically modified organism such as disease resistant plants. So coming to the history of genetics, so Walter Sutton in 1886 proposed that genes are located on chromosomes. Thomas Hunt Morgan discovered the first genetic mutant of fruit flies in 1910 and used fruit flies to unravel many details of transmission genetics. So Ronald A. Fisher, John B.S. Haldane, and Sewell Wright laid the foundation for population genetics in 1930s. Geneticists began to use virus and bacteria in 1940s. The rapid reproduction and simple genetic systems of these organisms showed a detailed study on organization and structure of genes. Then Watson and Crick, they described the three-dimensional structure of DNA in 1953, and in 1966 the chemical structure of DNA was determined, and Walter Gilbert in 1932 and Frederick Sanger in 1918 developed the methods for sequencing the DNA, and the polymerase chain reaction, a technique for quickly amplifying the DNA, was developed by Carey Mullis and the others in 1986. So in 1990, the gene therapy was used for the first time to treat human genetic disease in the United States, and the Human Genome Project was launched. In 1995, the first complete DNA sequence of Haemophilus influenzae was determined, and the first complete sequence of eukaryotic organism was reported a year later. This slide shows the gene therapy application and genetic engineering for the treatment of human disease. So as you can see this slide, there is a patient with genetic disease, and the cells of the patient are withdrawn, and the virus containing functional gene, the vector of that virus is inserted into the cells, and a new or correlated version of a gene is added to the cell, usually with the use of genetically engineered virus. The cells are then grown in the lab conditions and tested for their viability and functionality. And finally, it is implanted into the patients to cure the genetic disease. Coming to the future of genetics, genome sequences of many organisms are added to DNA databases every year, and new details about gene structure and function are continually expanding our knowledge of heredity. In future, the focus of DNA sequencing will shift from genome of different species to individual species. The new genetic microchips that are developed are used for analysis of thousands of RNA molecules, which has the activity of thousands of genes in a given cell. So microchips will have the information of thousands of genes in the given cell. So this way we can identify or detect any mutations occurred in the genes. So genetic information requires the continuous development of sophisticated computer programs to store, retrieve, compare, and analysis of the genetic data. So it has given rise to the field of bioinformatics, genomics, proteomics, and all these fields. The use of genetics in agricultural, chemical, and healthcare will continue to expand. This wide scope of genetics will raise significant ethical, social, and economic issues. Coming to the basics of genetics. So this everyone might be knowing. So cells are of two different types, eukaryotic and prokaryotic. So prokaryotic cells do not have a nuclear membrane, and they possess no membrane bound organelles, whereas eukaryotic cells are more complex, where they have a well defined cellular organelles such as chloroplasts, mitochondria, Golgi apparatus, and a gene is the fundamental unit of heredity. So this is a precise way in which a gene is defined often varies. So gene is a unit of information that encodes a genetic characteristics. So gene only is responsible for each and every character changes in a person or disease transmission from one generation to another. So genes come in multiple forms of alleles. A gene that specifies a characteristics may exist in several forms called alleles. For example, the color of a organism or an animal that changes from one progeny to another is mainly dependent on genes. So genes encode phenotypes. Important concept in genetics is distinction between traits and genes. So traits are not inherited directly, but genes are inherited, along with environmental factors determine the expression of traits. The genetic information that an individual organism possesses is an genotype, and that its trait is an phenotype. For example, in humans, the A blood group is a phenotype. The genetic information that encodes the blood type A antigen is a genotype. So next basics is the genetic information is carried in DNA and RNA. So as Watson and Crick they derived the structure of DNA. So they have also told that nucleic acids are present, and those nucleotides such as adenine, guanine, cytosine, and thymine. These are the four nucleotides which are present in RNA and DNA that gets altered. When it gets altered, we get mutation in the progeny. So the nitrogenous bases in DNA are of four types: adenine, cytosine, guanine, and thymine. And that of RNA are adenine, cytosine, guanine, and uracil. So next basics is the genes are located on chromosomes. The vehicles of genetic information are chromosomes that consist of DNA and associated proteins. The cells of each species have a characteristic number of chromosomes. So bacterial cells normally possess a single chromosome, whereas humans have 46 and the pigeon cells possess 80. So the number of chromosomes varies from organism to organism, and they play a very vital role. So the process of mitosis and meiosis ensure that each daughter cell receives a set of organisms' chromosomes. Mitosis is the separation of replicated chromosomes during the division of somatic cells, and meiosis is pairing and separation of replicated chromosomes during the division of sex cells to produce the gametes. So next, genetic information is transferred from DNA to RNA and RNA to protein. So this is the central dogma that everybody knows. Many genes encode the traits by specifying the structure of proteins. Genetic information is first transcribed from DNA to RNA, and RNA is translated into amino acid sequence of a protein. Mutations are permanent heritable changes in a genetic information. So gene mutations affect only the genetic information in a single gene. Chromosome mutations alter the number of structure of and therefore usually affect many genes. So any changes in the nucleotide sequence will lead to mutation. There are so many types of mutation. Some traits are affected by multiple factors. Some traits are influenced by multiple genes that interact in complex ways with environmental factors such as height. For example, if height is affected by hundreds of genes as well as environmental factors such as nutrition. So it also depends on genes and it also depends on nutrition. Evolution is a genetic change. Evolution can be viewed as two-step process. First, genetic variation arises, and second, some genetic variants increase in frequency, whereas other variants decrease in frequency. So coming to the cell cycle. So this diagram shows the cell cycle where the interphase, M phase, metaphase, the period of nuclear and cell division. So cell cycle is a life story of a cell, the stages through which it passes from one division to the next. So first, during the G1, the cell grows, and the cell may enter G0, a non-dividing phase. After the G1 or S phase, there is a checkpoint. So if the cells are not eligible, then it will not go further. The cell is committed to dividing in a S DNA duplicates. Then in a G2, the cell prepares for mitosis. After the G2 checkpoint, the cell can divide. So there is another checkpoint at the G2 level called the M checkpoint, and mitosis and cytokines take place in M phase. This process is critical to genetics because through cell cycle, the genetic instructions for all characteristics are passed from the parent to the offsprings. A new cycle begins after a cell has divided and produced two new cells. A new cell metabolizes, grows, and develops. At the end of the cycle, the cell divides to produce two cells, which can then undergo additional cell cycles. And this table shows the features of a cell cycle. So G0 phase, the major features are stable non-dividing period of variable length. And in interphase, we have three different phases: G1, S, and G2 phase. So G1 is responsible for the growth and development of the cell, and there is a S checkpoint. And S phase is the synthesis of DNA. G2 phase In G2 phase, the cell prepares for division. And there is a M phase where we have prophase, where chromosome condenses and mitotic spindle forms. And pre-metaphase, where nuclear envelope disintegrates, spindle microtubules anchor to kinetochores. And then we have the metaphase, anaphase, telophase, and cytokinesis. So the number of chromosomes and DNA molecules changes in the course of cycle. The number of chromosomes per cell equals the number of functional centromeres, and the number of DNA molecules as per the cells equals the number of chromatids. So as you can see that in different stages of cell division, in G1 you can see that there are 4 set of chromosomes, and in the prophase and metaphase it starts to divide, and in the anaphase we have 8 set of chromosomes, and the number of DNA also increases to 8 in the anaphase, and then it again reduces, it gets separated, and we again have 4 number of the centromeres. Coming to the next important topic called PCR. PCR is polymerase chain reaction. So it is a technique which is used to amplify a DNA. So it is mainly used in the criminology department to identify the criminals. So they get traces of samples such as hair or any blood, and that will be taken to the lab for analysis. So the DNA will be extracted from that sample, and it will be gone through PCR to identify the culprit. So, coming to the history of the PCR, how the PCR was discovered. So, in the early 1600s, Europeans arrived in the New World, devastating epidemics of tuberculosis to the Native Americans. So, anthropologists um, long argued that this disease was absent from the world before 1492, and Europeans first transmitted tuberculosis to the Native Americans. But on the basis of tuberculosis like lesions found on the skeletons and mummified remains predate European contact, they suggested that this disease was present in Native Americans before the European contact. So many diseases and even bacteria can gain access to the body after death, and that also can lead to some marks. So this idea was remained controversial. So in 1994, a pathologist named Arthur C. Offerhead and molecular biologist Wilmer Sallow teamed up to resolve this controversy. So Offerhead obtained access to the remains of the woman who died and was naturally mummified in Peru about 1,000 years ago and hundreds of years before. So they collected samples from that mummy, and they did the analysis. Um and the molecular biologist Sallow, he did the analysis of that sample, and he amplifies the sequence of DNA to search the sample from from the lady, and he found that it was Mycobacterium tuberculosis. So when he matched that sequence with the modern day Mycobacterium tuberculosis, it showed a copy of 97 base pair fragment of DNA. So Sallow detected an identical 97 base pair piece of DNA by applying PCR to the DNA samples from the lungs and lymph tissues. And then they found and they identified that Europeans were still the most likely cause of tuberculosis epidemics that accompanied their arrival in the Americas. So this story of discovery of tuberculosis in the remains of 1,000-year-old woman illustrates the power of PCR in the molecular biology. So coming to recombinant DNA technology, so it is a set of technique for isolating, altering, and studying the DNA segments. The term recombinant is used because frequently the goal is to combine DNA from two distinct sources. So genes from two different bacteria might be joined. For example, or a human gene might be inserted into the viral chromosome. So commonly called genetic engineering. DNA technology now encompasses an array of molecular techniques that can be used to analyze the DNA sequences. So, basic concepts of recombinant DNA technology. So, in 1973, a group of scientists produced the first organism with recombinant DNA. Stanley Cohen at Stanford University, uh, he gave colleagues, he inserted a piece of DNA into plasmid in another, creating an entirely new set of DNA molecule. Then they introduced the recombinant plasmid into E. coli cells. So, with a short time, they used the same method to stitch together genes from different bacteria. These experiments ushered one of the most momentous revolutions in the history of science. So, recombinant DNA technology has proved that new information about the structure and function of genes has altered many fundamental concepts. So, this slide shows the milestones in biotechnology and rDNA technology. So you can see that from 1958, DNA polymerase was purified, and then a complete set synthesis of in vitro discovery of first sequence restriction endonuclease, which are the molecular scissors. So in 1978, human genomic library was constructed. Then the development of polymerase chain reaction in 1985, and in 1988, the successful production of genetically engineered crops. And in 1989, first field test of genetically engineered virus. And in 1991, development of transgenic pigs and goats. And in 1995, Haemophilus influenzae genome was sequenced. And in 1997, human clinical trials of antisense drug and DNA vaccines began. And in 1998, first cloned mammal, sheep dolly, was. So coming to the rDNA technology, it is used to create a number of commercial products, including drugs, hormones, and enzymes and crops. So rDNA is used to cure the cancer, diagnostic, genetic, produce drugs. So the basic problem is with the gene is are minute and they are very difficult to identify and screen. Even when viewed with a powerful microscope, we can see a DNA only like a thread-like structure. So gene expression is a complex process requiring a number of DNA sequence elements, some of which lie outside the gene itself. So molecular techniques have been identified to overcome all of them. Human genes are routinely transferred to the bacterial cells. So recombinant technology has been used to create genetically modified crops. So as you can see in this image, so it is a genetically modified corn that produces a toxin that kills the insect. So there is no need to spray the pesticides on these or insecticides on these crops. So it is a genetically modified. So in recombinant DNA, the following techniques are to be implemented: methods for locating specific DNA sequence, techniques for cutting DNA at precise location, procedures for amplifying a particular sequence billion times, producing enough copies of DNA, methods for mutating and joining DNA fragments, procedures for transferring DNA sequence into the recipient cells. So first we come to the cutting and joining of DNA fragments. So for cutting and joining of DNA, we need a specific tool called the scissors, molecular scissors. So molecular scissors are nothing but they are the restriction enzymes, also known as restriction endonucleases, that recognize and make double-stranded cuts in a sugar phosphate backbone of DNA. These enzymes are produced naturally by the bacteria where they are used in defense against viruses. In bacteria, restriction enzymes recognize particular sequence in viral DNA and then cut it up. A bacterium products its own DNA from restriction enzyme. So when a bacteria is in the synthesizing an enzyme, so that enzyme may affect its own cell also. So they have a mechanism called methyl groups. So to their DNA, they have a methyl group added to it, and that will not let the cleavage of its own DNA. So three types of restriction enzymes have been isolated from bacteria. So number one is type 1, type 2, and type 3. So type 1 can cut the DNA at random sites where and that may be at a distance of 1000 base pair. The size of the DNA to be cut by type 1 are 1000 base pair or more. And for type 2, can identify the DNA and recognize the sequence, and they can cut 10 base pair length. And type 3 and can cut a DNA at nearby sites of 25 base pair. So the recombinant DNA is done with the type 2 restriction enzymes. So there are around 800 different restriction enzymes that recognize and cut DNA at more than 100 different sequences that have been isolated from bacteria. So many of these enzymes are commercially available. Examples of some commonly used restriction enzymes are BAM1, COF1, DRA1. You can see a table that is there beneath. It has EcoR1. So EcoR1 is a enzyme that is isolated or extracted from microorganism named Escherichia coli. And here you can find there is a recognition sequence, and they have given a sequence where it will cut at the cohesive end. Cohesive end means it will cut only a single nucleotide at one end, and the blunt end means it can cut three or four nucleotides at a time. So you can see that in the upcoming organism, you can find that in the blunt end, you can find there will be more nucleotides where the cut will take place. And in cohesive, the cut can take place at the first end or last end, but it will take place only in one nucleotide. So the sequences recognized by the restriction enzymes are usually from 4 to 8 base pair long. Most enzymes recognize a sequence of 4 to 6 base pair long. Most sites are palindromic. Palindromic means if you read from both the sides, it will be the similar, like Malayalam. So Malayalam, if you will, is a language. If you will read from both the sides, it will be same only. Some of the enzymes make staggered cut of the DNA. For example, HIND3 recognizes the following sequence. So HIND3 cuts the sugar phosphate backbone of each strand at the point indicated by the arrow. So you can see an arrow mark at the last nucleotide adenosine. So generating fragments with short single-stranded overhanging ends. So after the cutting is done, these ends are called cohesive ends or sticky ends because they are complementary to each other and can spontaneously pair with to connect the fragments. So thus DNA fragments can be attached together, and any two fragments cleaved by the same enzyme will have complementary end and will pair together. So their cohesive ends have paired two DNA fragments can be joined together permanently by the enzyme DNA ligase. So the enzyme that joins together these two nucleotides is known as DNA ligase. So which seals the nicks between the sugar phosphate group of fragments. So not all restriction enzymes produce staggered cut or sticky ends. So PVU3 2 cuts in the middle of the recognition site, producing blunt-ended fragments. So you can see in this nucleotide sequence, there is CAGCTG. And in the 3 prime, 5 prime, you can see GTCGAC. So it is making a cut at G, C and CG between that. So this is called as a ended fragments. The fragments with blunt end must be joined together in two ways, which will be discussed further. So the number of restriction sites is related to this figure shows the restriction reaction where they have done an experimentation. They have placed a DNA, purified DNA, in a small tube with a buffer solution, and a small amount of restriction enzyme is added along with it. The reaction mixture is heated at 37 degrees Celsius. So in few hours, the enzyme cuts all the restriction sites in the DNA, producing a set of DNA fragments. So you can see that in a linear DNA, they have given there are four digestion sites, and with the linear piece of DNA, the number of fragments produced is more than the number of restriction sites. Whereas in circular DNA, the number of fragments produced is equal to the number of restriction sites. So in the left, you can see an image where the restriction enzymes make double-stranded DNA cuts in the sugar phosphate backbone. So HIND3 is the name of the restriction endonuclease enzyme that makes staggered cut in the DNA. So by producing single-stranded cohesive ends, and PVU2 is a restriction enzyme such it cuts both the end of DNA. So as you can see that it is cutting the sequence into two half, whereas HIND3 is able to cut only in that specific region. So, and the next one is the DNA molecules cut the same restriction enzyme have complementary sticky ends, whereas the nicks in the sugar phosphate backbone of the two fragments can be sealed by DNA ligase. So again, to attach them, we need an enzyme called DNA ligase. So after the cutting and the cutting of the fragment by DNA is done, we go for the visualization of the band by electrophoresis technique. And electrophoresis, as we all know that we use agarose gel electrophoresis for visualizing the gels. So it is a technique in which we use electric charge to pass through the buffer solution. It contains tris HCl solution, and we have a tank buffer which when run in the vertical, as DNA is negative, the charge passes from negative to positive. So the cleavage of DNA by restriction enzymes, and in the B you can see migration of fragments. So the DNA moves from negative to positive. So as the charge is passing through, it will take the DNA and a separation will take place. So after electrophoresis, you can see that the large DNA fragments appear on the top, and small DNA fragments appear at the bottom of the gel. And gel electrophoresis can be used to determine DNA molecules. So D is the stained gel. So for staining of the gels, we use ethidium bromide. So ethidium bromide is an fluorescent dye that goes and binds with the DNA, and it will fluorescence when we visualize it in the UV, ultraviolet radiation. So we have instruments like UV transilluminator to visualize these DNA gels, and even in industries we have gel documentation unit where we can visualize these gels. And the original concentrated sample of purified DNA contain millions of copies of DNA, and thus each band represents millions of copies of identical DNA fragments. So this gel can not be used only for visualizing the normal DNA or the restricted DNA, but we can also use for RFLP and RAPD and PCR samples. So PCR done samples also can be visualized. Normal genomic DNA, these also can be visualized in the gel. So next, coming to the important tool named plasmid vectors. So plasmids are circular DNA molecules that exist naturally in the bacteria, and they contain some regions, specific regions called origin of replication. So one is origin of replication, where therefore able to replicate independently. And plasmids typically used in cloning have been constructed from larger naturally occurring bacterial plasmids. So PUC19 is a plasmid, and it is a typical cloning vector where we have different enzyme cleaving sites. So these enzyme cleaving sites will cut at a particular region, and our DNA of interest will be inserted into that plasmid, and then it is inserted into the bacteria for transformation studies. So ampicillin is an antibiotic that normally kills bacterial cells, But any bacterium that contains PUC19 plasmid will be resistant to antibiotic because it is containing a antibiotic resistant gene. So LAC-Z gene encodes the beta galactosidase, which normally cleaves lactose to produce glucose and galactose. The enzyme will also cleave the chemical called XGAL, producing blue substance. The XGAL is placed in the medium, and these three characteristics of idealized cloning vector. So first is a cloning vector must contain an origin of replication recognized in the host cell so that it is replicated along with the DNA. And then we have it should be selectable markers. So trait that enables containing vector to be selected. And then a cloning vector needs single cleavable cleavage site for one or more restrictions enzymes. So PUC plasmid is a typical cloning vector. So it contains a cluster of restriction sites such as ORI called origin of replication and two selectable markers, and it is ampicillin resistance gene called LAC-Z gene. So it contains a LAC-Z gene which is resistant to ampicillin. And on the right hand side you can see a image of PBR322 plasmid. So the map is marked off with 105 Dalton. The location of some restriction enzyme are indicated. So this plasmid is resistant to both ampicillin and tetracycline. So it has both APR and tetracycline resistant genes. So whenever we are growing this plasmid in laboratory for scale up, we need to add ampicillin and tetracycline in the media as it is ampicillin and tetracycline resistant. So once the fragments have been isolated, they are ligated with an appropriate vector, such as plasmid, to form a recombinant molecule that produce in the host cell. So as you can see that there is a plasmid on this diagram. The plasmid is cut with the ECO R1 enzyme, and then the complementary base pairing is happening, followed by ligation. So in ligation, the recombinant DNA molecule is created after the ligation. So the new gene is getting inserted into the plasmid. So that is the donor DNA. That is the DNA of a host. So that host DNA is getting inserted into our plasmid, and it is forming a recombinant DNA. So and it is inserted into the E. coli, and that is called transformation. So we use calcium ions for the transformation process, and the culture bacteria in the medium is added with tetracycline. That tetracycline will we are adding because it is tetracycline resistant gene. It is having a tetracycline resistant gene in the plasmid. So we need to give that tetracycline, otherwise that bacteria will not grow. So tetracycline resistant colony is transformed on the plate. So finally you can see a plate that contains a tetracycline resistant colony. So if the growth of bacteria is found on the plate, it is clear that the bacteria is transformed, and it is ready for the scale up. So recombinant plasmid construction and cloning. So for construction and cloning, the scale of sticky end of fragment and plasmids is enlarged to illustrate the complementary base pairing. The electron micrograph shows a plasmid that has been cut by a restriction enzyme and a donor DNA fragment. So you can see a TEM image, a micrograph image where the plasmid is cut and its image is shown as a thread-like structure in that black background in that image. The micrograph shows a recombinant plasmid. Coming to the next diagram, it is a diagram that shows the transformation of bacteria and the the membrane. They have used a membrane called nitrocellulose membrane to transfer the colonies from the plate to nitrocellulose membrane, and they add a cDNA probe to check the dark spots on the film that indicates that our colony is a recombinant. So first they have taken a plasmid which contains both ampicillin and tetracycline resistant gene, and its restriction cleavage with the endonuclease enzyme. They have cut the DNA, and the foreign DNA also they are parallelly doing the restriction enzyme cleavage, and that they are ligating with the plasmid, and it is transformed into the bacteria. So then after it is transformed, it is plated on the agar plate media containing the antibiotic. So the it is then transferred to nitrocellulose paper, and after transferring it is lysed, and the DNA is denatured. So cell lysis takes place so that all the genetic material inside the cell will come out on the nitrocellulose membrane, and to that they add a complementary DNA probe, which contains a fluorescent probe. So then it is washed off with the unhybridized cDNA, and in the radiography they can see the fluorescence of phosphorus 32 enabled fluorescence, and this shows the recombinant DNA technology in which they have used bacteriophage. So first they have taken the cellular DNA and they have cleaved it out, and the bacteriophage DNA. Then they have cleaved the DNA with restriction enzyme parallelly, and the fragments are ligated so that they both combine and form a recombinant DNA. So that recombinant DNA is ligated into the in vitro phage. So the hybrid phage mixture contains the recombinant DNA. So that phage is again cultivated on the plate to see whether it is formed, the DNA is formed properly or not. So to check that only we add antibiotics. So isolation of phage clones. Then isolation is done on the nitrocellulose filter, where we add radioactive probe and incubate. So after incubation, the auto radiograph, we can see that there is on the thin film there is a clone activity formed on the radiograph, that nitrocellulose membrane, and when that nitrocellulose membrane is placed on the Petri plate, the desired clone gives fluorescence. So the mRNA is not available in sufficient quantity to serve as a probe. The desired mRNA species can be converted into cDNA by reverse transcription. So previously cloned gene or portion of gene can be used as probes. So this approach is effective when there is a reasonable amount of similarity between nucleotide and two genes. So often P32 is added to both the strands so that the radioactive strands can be located with autoradiography. So here they have used lambda phage as a vector. So the preparation of genomic library, each colony in the bacterial lawn is a recombinant clone carrying a different DNA fragment. These are some of the recombinant DNA cloning vectors, and these are the uses of those vectors. So first one is plasmid E. coli, so PBR322. So PBR322 has different restriction enzyme such as BAM H1, ECO R1, HEM3, HIND3, PST1, SAL1, and ZOR2. So these are all the different restriction sites where the enzymes can go and cut and the new DNA can be inserted. So it carries genes for tetracycline and ampicillin resistance. So, and we have some yeast artificial chromosome. So the host is yeast and carries a gene for ampicillin resistance. So multiplies in Saccharomyces cerevisiae. Then we have some viral based cloning vectors. So they also have ECO R1, HIND1, BAM1, and SST1. And some viruses have only single restriction sites such as ECO R1, and some only have BAM H1, and plasmid such as TI plasmids. So they have SMA1 and HPA1. So it is used in the maize plasmid for plant cultivation. So applications of genetic engineering. The production of medicinally useful proteins such as somatostatin, insulin growth hormone, and interferon is of great practical importance. So interleukin 2, it is a cytokinin, and blood clotting factors 8 have been recently cloned and undoubtedly other important peptides will be produced in the future. A particularly interesting development of is the use of transgenic corn, as we saw in the earlier slides, and soybean plants to produce monoclonal antibodies. So monoclonal antibodies are nothing but they are uh, a set of antibodies which are produced in a mice. They are injected and they are provoked in the mice, and mice cells produce these monoclonal antibodies, and they are also having so many therapeutic values. Genetically engineering mice now can be produced fully human monoclonal antibodies. So synthetic vaccines, for instance, vaccines for malaria, rabies, are all developed with recombinant techniques. So probes are now being used in the diagnosis of infectious diseases. An individual could be screened for mutant genes with probes and hybridization techniques. A type of genetic surgery called somatic gene therapy may be possible for afflicted individuals. For example, cells of an individual with a genetic disease could be removed, cultured, and transformed with cloned DNA containing a normal copy of defective genes. So, coming to the next topic, it is gene expression and kitty boo colors. So, as we have seen a firefly in winters in the village areas, like rural area, we find some light glowing insect. So, it is nothing but it is a beetle that has a luciferase gene in its DNA, and that gene is responsible for the production of that light. So, genetic engineering techniques have performed with luciferase genes, and there is a scientist named Keith Wood who has cloned the genes and inserted them into E. coli. So when the bacteria are exposed to luciferin, they glow. So he has cloned the genes and inserted them into the E. coli. So you can see different colors of expression in different streak they have done. So when it is exposed to luciferin gene. So here are some of the important human peptides and proteins synthesized by genetic engineering. So as you can see, blood clotting factors 8, it is useful in the treatment of hemophilia, and calcitonin, it is useful in the treatment of osteomalacia. Osteomalacia is a disease where they have their bones become affected. So the bone marrow cells get affected and it becomes the soft bones. And then there is a condition called anemia. So for anemia, they are using erythropoietin and growth hormone. For growth hormone, also they have genetically modified. And relaxin, it is a peptide which is used in the childbirth. And there are some somatostatin for treatment of acromegaly, and streptokinase, which is used as an anticoagulant. So anticoagulant is very essential in the medical field, as we are having some chemical which we traditionally use, such as heparin and EDTA. So these are commercially used in as anticoagulant. So now we have tissue plasminogen activator. So these peptides, when they are introduced into our human body, they act as an anticoagulant. And tumor necrosis factor. So coming to the probes, probes are now being used in the diagnosis of diseases. An individual could be screened for mutant genes with probes and hybridization techniques. So genetic surgery called somatic gene line therapy may be possible for afflicted individuals. So cell individual with a genetic disease could be removed, cultured, and transformed. So these cells, when reintroduced into the cell, they become established. The expression of the normal genes might be cure the patient. So coming to the pharma applications. So I have worked in a pharma company where we were working with ursodeoxycholic acid. So it is also known as ursodiol. It is a secondary bile produced in humans and most other species for metabolism by intestinal bacteria. So it is a medication used in the management and treatment of cholestatic liver disease. So the short form of urodeoxycholic acid is UDCA. So we call it as UDCA. So UDCA reduces the elevated liver enzyme by facilitating bile through liver and protecting the liver cells. So in the pharma company, we were a group of people who were working with the conversion of lithocholic acid to ursodeoxycholic acid. So lithocholic acid is a substrate that we give to the bacterial cell. So and the bacterial extracellular enzymes such as reductase and monoxidase. So these are the two different types of enzymes that enable the conversion of lithocholic acid to ursodeoxycholic acid. So for that process, we normally do a process called transformation. So we take a cell and we do incubation for some period, and then we give a heat shock so that the plasmids and cells together will ligate inside and will get inside and it will transform. So that is called a biotransformation experiment where we insert a particular gene carrying plasmid into the bacterial cell. So that will give us a recombinant DNA, and we check the cells on the plates. We actually plate the agar plates, and we add ampicillin and streptomycin resistant. As our plasmid is ampicillin and streptomycin resistant, so we have added these two antibiotics for the growth. So not even in the plates, we also, when we scale up in broth in higher volumes from 10 to 20 or 30 or even 100 ml, we go for adding the antibiotics. So first we make the competent cells. So what is meant by competent cells? So these are the cells which are prepared to receive the plasmid. So it is called competent cell preparation. So we take the normal bacterial cells, we centrifuge it, and we take the pellet, and then we resuspend in calcium chloride and incubate it on ice. So we give a cold stress to that organism, and it then we wash it with and resuspend in transformation buffer. So this leads the calcium ions to go and adhere to the cell wall, and then it will let the plasmid to easily get inside the cell. That's called a chemical transformation. So bacterial transformation, first we prepare the competent cells that I have told you in the previous slide, and then the plasmids get inside the cell, and then we it gets recovered, and for confirmation we plate it on the agar media containing the respective antibiotics. So this is another product which with on which we were working. It is pregnenolone. So it is a steroid hormone that plays a key role in the production of other steroid hormones such as progesterone, DHEA, and estrogen. The amount of production in the body is very less, so we give it as a supplement. So pregnenolone is used to sharpen the memory, reduce stress, stimulate the immune system, promote detox, prevent heart disease, and slow the aging process. So for this, we use Mycobacterium smegmatis as a host, and we transform the desired plasmid into the Mycobacterium smegmatis, and we do the cultivation of the cells in laboratory in shake flask. Then after the cultivation is done, we do the induction of uh, the cells with acetamide. So that acetamide will lead to the production of some enzymes that help in the bioconversion of our substrate. So here we are using a substrate as phytosterol. So phytosterol is a plant-based substrate that is given to the bacterial cell after the growth is done and they are matured, and then it is gone for the multiplication, and after it has reached the particular pellet volume, so we check the absorbance, and we also routinely check the what is that biomass. So when the biomass is reached to that particular extent, we will take it further for the bioconversion experiment where we will add the substrate called phytosterol. That phytosterol will get converted to pregnenolone. So we are checking different optimization parameters to increase the conversion from phytosterol to pregnenolone. And this I already told you in the previous slide, lithocholic acid to urodeoxycholic acid. So the difference in these two experiments is that after the growing the cells, we go for the lysis of the cells. And we use the enzyme, crude enzyme for the bioconversion. We also use the purified enzymes. So we follow the techniques such as ultrafiltration, diafiltration, SDS page to visualize the amount of protein expressed, and then we go for ion exchange chromatography to purify the enzymes, and that purified enzymes we take it for the bioconversion study. And finally we do the TLC, thin layer chromatography, to check whether our desired substrate has got converted to the product or not. And this is the research and development laboratory of my previous organization where I have worked. So this first one is the lobby where we enter. It is called crossover bench. So we remove our dress and we go inside. Why there is so much of safety and security is it is all to be done in a aseptic condition. The microbes from outside should not go into our media, and whatever is work is done inside, that should not come outside. So this is called a aseptic condition, where we have HEPA filters installed on the culture rooms. And as you can see on the second figure, there is a pass box, a square pass box, which is there on the wall. And that pass box is useful to take the things from outside to inside the biosafety chamber. So we work inside a biosafety chamber in a suit like this. This is called gowning. So whenever we are going into a biosafety cabinet, we have to wear this gowning, and then only we will work with the microbes for the molecular biology or any of the microbiology experiments. And this is a fermenter where we do the scale up of our desired protein or desired bioconversion studies also we perform in these. And we also do some antibiotic studies. And this is a culture handling room, and this is a HPLC analysis room where we do the final analysis of the converted product. So coming to the final slide, recombinant DNA techniques are playing an increasingly important role in research on the molecular basis of disease. The transfer from research to practical application often is slow, because it is not easy to treat a disease effectively till its molecular mechanism is detected. So genetic engineering may aid in the fight against disease by providing new information about its nature, as well as by aiding its diagnosis and therapy. So thank you very much.
Any doubts asked to sir? Thank you, sir, for your wonderful and valuable presentation on exploring current challenges in genetic engineering and advanced technique in medical science. Thanksgiving is a time to give, time to love, and a time to reflect on the things that matter most in life. I deem it my pleasure to invite M. Ashwini, Assistant Professor, Department of Biochemistry, to deliver the vote of thanks.
Good morning to one and all present here. Gratitude can transform common days into thanksgiving, turn routine jobs into joy, and change ordinary opportunities into blessings. It's my great pleasure to propose vote of thanks on this wonderful moment. I deemed it pleasure to thank Reverend Mother C. Noel Rani, FSAG, the Secretary, who is always behind us in all the accomplishment of our institution and made this event a special one through our presence. Thank you, Mother. I feel too proud to thank our beloved principal, Reverend Dr. E. Gilita Sumangali, FSAG, who has given this opportunity and guidance to proceed over the program in a great manner. We will ever remain grateful to you and we thank you for being with us today. Thank you, sister. I would like to thank our Vice Principal, Reverend Dr. A. John Diana, FSAG, for all words of wisdom that radiated source of energy within us. Thank you, sister. Today, my words are not enough to express the gratitude. On behalf of our college, I would like to thank our chief guest, Dr. Santosh Shigamani, Team Lead Product and Process Control at Pasha Bioscience Private Limited, who graced us with his thought-provoking address set on genetic engineering advanced in medical science, a perfect platform for our college. Thank you, sir. We are all inspired by your great words.
Thank you, madam. Thank you.
I show my sincere thanks to Mrs. J. Sumadi, Head of the Department Biochemistry, for her perfect logistic support and guidance towards the staff members and students of our department. And she is one who is always behind us in all the deeds of our department now and then. Thank you, ma'am. Finally, a note of thanks to all the staff members and lab assistants of our department who have been source of strength to us through their support and various other kinds of help. Thank you, ma'am. I thank the staff and student participants of our college and from various colleges for bringing grace to the webinar. Thank you all. My thanks will not complete if I do not acknowledge my dear student committee. Each one of them with love reached out to make this a great one. Thank you, my dears. Once again, I thank you one and all. Now wind up the program.